Thank you very much, Bridget, for a lovely introduction, and thank you very, very much to the Physiological Society for the honour of providing this lecture. And as I say, I feel a bit of an interloper here as a biochemist by training, but hopefully over the course of the next 40 minutes or so, I'll lead you on a journey from the molecular mechanisms of neurodegeneration, which has really been the focus of my research for over a decade now, to physiology and cellular physiology and organismal physiology, and then back again to disease. And the focus of this is going to be Leucine Rich Repeat Kinase 2. And I thought actually it's appropriate with a lecture of this type to begin at the end. And I'd like to start by thanking all of the people I've worked with over the last 10 to 15 years now, working on LUC2, in particular my collaborators and students and postdocs at the University of Reading, in particular Claudio Manzoni, who's worked with me for almost a decade now, and my collaborators at UCL, the NIH, the University of Padova, and the Francis Crick Institute, and collaborations with these groups have resulted in the data that I'll be presenting today. And so in many ways, I feel that I'm receiving this prize not for myself, but as a representative of a group collaboration. And as Bridget said in her introduction, the origins of the Bayliss Starling Prize really come from Gow Street and University College London and the work of Bayliss and Starling. And you can draw a slightly wobbly line between Bayliss and Starling to some of the other giants of physiology and previous recipients of the Bayliss Starling Prize, such as J.Z. Young, A.V. Hill, and Bernard Katz, all of whose work was carried out at University College London. And as a naive PhD student, I recognize these names not so much for their massive contributions to science, but as the lecture theatres to which I visited and occasionally attended lectures. But actually, my own connection to UCL comes through the work of two individuals, both Johns. John Hardy over here on the left of the slide, and John Collins, my PhD supervisor at the MRC Prion Unit, both of whom are now at UCL. And this actually provides quite a nice link towards the starting point for this today's lecture, as both are leading exponents of the application of neurogenetics to understanding neurodegenerative disease, contributing to Alzheimer's disease and Parkinson's disease in the case of John Hardy. And he won the Breakthrough Prize a couple of years ago, which he got this statuette for, which I thought is rather nice, and he keeps on his desk. And the prion diseases in the case of Professor Collins. And so as I say, what I'll do is I'm going to start this lecture looking at a disease, and in particular Parkinson's disease. We've known about Parkinson's disease actually for over 200 years now. It's just last year was the 200th marking point of the original description of Parkinson's disease by James Parkinson and his essay on the shaking palsy. James Parkinson, incidentally, is one of the few people I know who has a disease named after him, but also has a family of fossils named after him. He was also a very eminent geologist. And Parkinson's disease is illustrated by this video here, which is one of the earliest film footage we have of a Parkinson's patient from the early years of the 20th century in a hospital just outside of Brussels in Leuven. It is a movement disorder. It's a neurodegenerative movement disorder, primarily of old age, and is characterized by tremor, rigidity, and slowed movement. And this is all very visible in this video. And if you look very carefully, you can note that there is a tremor of the right hand. And these are the symptoms that we recognize now as being associated with the movement side of Parkinson's disease. And we've understood the pathological basis for Parkinson's disease for many, many years. In the 1920s, with the work of Frederick Louis, we began to understand that a particular region of the brain, the substantia nigra, was specifically affected by degeneration, by loss of cells, by the accumulation of intracellular aggregates, which are now called Lewy bodies after Frederick Louis, which we now know are made up of an aggregated protein called alpha-synuclein, which was discussed a little bit in this morning's excellent lecture on the glymphatic system, talking about the clearance and removal of aggregated proteins in the brain. And from the work of Arvid Carlsson, who died earlier, sadly passed away earlier this year, the key role of dopamine in the disruption to the systems in the brain that were associated with Parkinson's disease was recognized in the 1950s and 1960s, which led to the development of a host of drugs which are still to this day used in Parkinson's. And the frontline treatment for Parkinson's remains a drug that has been in use for over 50 years. And this really speaks to one of the key problems that we have with Parkinson's disease, and this extends actually to most neurodegenerative diseases, that we understand some of the neurochemical basis, we understand the systems which are being disrupted, but we don't understand why those systems are being disrupted. What is causing the cells in the brain, in the regions that are affected, to die? And this is something that has changed over the last 20 years with the application of first familial genetic analysis, looking at rare families who carry inherited mutations in neurodegenerative disorders and in Parkinson's disease. And more recently with the advent of genome-wide association studies, which have allowed us to look at common variation across the human genome to understand what's going wrong in Parkinson's. And this is a summary that's up to date as to earlier this year of the genes that have been implicated in Parkinson's disease. And as you can see, we now have an extensive list of genes where coding mutations or loss of function mutations result in dopaminergic cell death in the substantia nigra and result in the symptoms that we recognize as Parkinson's. And it's hoped that by understanding how these genes are affected, we can get an insight into the molecular mechanisms underpinning Parkinson's disease, the physiological consequences of those mutations, and develop drug treatments. And actually, what I'll be talking about today actually leads through all the way to translational neuroscience. So we're now beginning to put together a genetic architecture for Parkinson's disease, leading from causative mutations, which are shown here as very rare in red at the top left-hand side of this slide, where if you have a coding mutation or a loss of function mutation, you will develop a disease, all the way through to much more common variation that is present widely in the human population, which will very, very slightly increase the risk of you developing Parkinson's. What I'll be focusing on for the rest of this talk is one of those genes that has come out of studies of Mendelian disease, located up here in the top left-hand corner, but has also been identified by genome-wide association studies, suggesting that dysfunction of this gene and its protein product are also linked to the more common idiopathic form of disease. And this gene is Leucine Rich Repeat Kinase 2. So mutations in LRRK2 were first described back in 2004 by two studies published by a German and American group and a British-American-Spanish collaboration. And they identified a series of mutations, coding changes in the LRRK2 gene, which were linked to autosomal dominant Parkinson's disease. One of the key things that came out of these original studies was that mutations in the LRRK2 gene were interesting for a range of different reasons. One of the key observations in the first paper was that it was associated with pleomorphic pathology. One of the themes that I'll return to through this lecture is the idea of both of pleomorphic impacts in terms of cellular pathology, but also pleiotropy, and the ability of variations in genes to have multiple effects, some of which may be directly related, some of which may be more distantly associated. And Dennis Dixon, who's one of the leading neuropathologists in the world, actually described LRRK2 as potentially a Rosetta Stone for neurodegeneration. If you look in the brains of individuals with LRRK2 mutations, they possess the Lewy body pathology, which we normally ascribe to Parkinson's disease, and indeed is required for a diagnosis of Parkinson's, shown up here at the top of the slide. But some of the members of the families don't present with Lewy body pathology. They present with tau pathology, more associated with progressive supranuclear palsy and with Alzheimer's disease, shown here in the center. And some members of these families also present with TDP43 pathology, which is associated with frontotemporal dementia and with amyotrophic lateral sclerosis and motor neuron disease. Sometimes these pathologies are seen in the same family with the same mutation and the same clinical phenotype, but when you look in their brains, there are very different things going on. And this actually is a puzzle which we've struggled to understand and find an answer to this very day. LUC2 is a very challenging gene to work with for a number of different reasons. One of the key reasons being that it's a very large gene. It's got 51 exons, and it codes for a protein of 2,579 amino acids. What's shown here is an artist's impression, if you like, of what we think the LUC2 holoprotein looks like. We think that it forms a dimer in solution, and we think that it is active in a trans way, communicating within the molecule to try and progress signal transduction. It's a multi-domain protein. It contains multiple functional domains. And so in a way, speaking back to the idea of pleiotropy and multiple outputs for a single gene, here is a key example of a gene which possesses multiple activities within one open reading frame. It has several protein-protein interaction domains, indicated here by ankyrin repeats, leucine rich repeats, and a WD40 domain, but critically, two enzymatic domains, a ROC GTPase domain located in the center of the protein, and a kinase domain located towards the C-terminus. And importantly, mutations causing Parkinson's disease are actually scattered throughout this protein. It's a member of a very interesting group of proteins called the ROCO proteins, which share this multi-domain, multi-activity function. And indeed, it's one of only 3 proteins in the human genome, human proteome, that possess both a GTPase activity and a kinase activity in the same open reading frame, making them providing potentially fascinating insights into the way in which cells control signal transduction. And this is very much an area of research that I've been actively involved in the past 5 or 6 years. The pleiotropy associated with LUC2 extends beyond Parkinson's disease, and one of the unexplained aspects of LUC2 biology and its physiological role is how it can be involved in a multitude of different disorders. We know it most notably for its role in Parkinson's disease, where it's involved both as a risk factor and as coding variants in Mendelian disease, and has been described as a pleiotropic gene from that aspect. But it's also been identified in inflammatory bowel disease as a potential risk factor. Coding variants in LUC2 are also associated with an increased risk of cancer, and indeed individuals with Parkinson's disease who have coding mutations in LUC2 also have an increased risk of certain types of cancer. It's also, most intriguingly, linked to leprosy, and a genome-wide association study published almost 10 years ago now indicated that variants around the LUC2 genetic locus are involved in increased risk for multibacillary leprosy. And one of the key challenges of understanding what LUC2 is doing, both in Parkinson's and across all of these disorders, is trying to understand how one gene can have such a varied effect in multiple human disorders. As I said, LUC2 is a dual-activity enzyme. It has a kinase and GTPase domain. And the early research that I was involved in at the NIH, and I was basically listed on the consumable budget at my boss's lab, Mark Cookson's lab, which is where the consumable point comes from, focused on trying to understand how these mutations alter the activity of LUC2 and change the biology of the cells within which it resides. And to do that, we took a model approach using the RAS GTPases as an analogy to the way in which we thought that LUC2 might be functioning. And this was previously been proposed for LUC1, a closely related protein to LUC2. The idea being that LUC2 as a multi-domain and multi-activity enzyme cycled between a GTP and GDP bound state here, controlling a kinase activity, which was linked to disease. And Elisa Grigio, a former colleague and collaborator of mine, carried out a series of experiments to look at the enzymatic activity of LUC2, focusing on the kinase domain of LUC2, demonstrating that the most common mutation in LUC2, the G2019S mutation, increases kinase activity, work that was published around the same time by a number of other groups, including the Dawson's group at Johns Hopkins University. At the same time, I was involved in knocking out the kinase activity of LUC2 using site-directed mutagenesis to remove the activity, and demonstrated that the kinase activity of LUC2 was exquisitely important in cytotoxicity observed with these mutations, data which is shown over here on the right-hand side of the slide, with cytotoxicity observed with coding variants, which is partially alleviated on a kinase-dead background. And actually, this was a very important observation and has led, over the course of the last 10 years or so, to, and based on the work of a number of other groups looking at the kinase activity of LRRK2, to the development of a host of small molecule inhibitors of LRRK2 function, which have now progressed to the point where they're going into clinical trials. And Denali Therapeutics over in California are now testing kinase inhibitors trying to modulate LRRK2 function in healthy individuals. So it's now entered phase 1 trials, showing how we can move from a molecular understanding, and actually I'll be talking a little bit about the physiological impacts of LRRK2 biology over the coming slides, all the way through to translational neuroscience, which I think speaks very much to the point that Bridget was making at the beginning of this lecture. My own role in all of this was very much focused on the GTPase activity of LRRK2. And what my work and work of a number of other groups around the world has now demonstrated is that mutations in LRRK2 can also act to modulate and reduce the GTPase activity of LRRK2, data which is shown over here, showing the wild-type protein and the impact of mutations in the ROC domain, shown here in red, which essentially reduces the activity to a background level. What we also found was that by modulating the GTP-GDP occupancy of the ROC domain, the ROC-GTPase domain, you could also influence the kinase activity of LRRK2, data which is shown over here on the right-hand side by autoradiography. And so what began to emerge was a picture of multiple enzymatic activities, pleiotropic enzymatic activities, interacting with one another and interacting in a disease situation. Working with Junpeng Deng, a structural biologist at the Oklahoma State University, we also were able to elucidate a crystal structure for the GTPase domain of LRRK2. And to date, this is one of the few bits of structural information, angstrom resolution structural information that anybody has been able to generate for LRRK2. What we found was that the mutations that cause Parkinson's actually sit at the interface of a dimeric structure for the ROC domain of LRRK2. Going on to look at this using biophysical characterization and moving into solution biology, using a combination of differential scanning telemetry, shown over here on the right, and circular dichroism spectropolarimetry, we were able to show that the mutations in LRRK2 actually destabilize the GTP and GDP-bound state of LRRK2, moving from a genetic study through a molecular cellular study to understanding the biophysical basis of the impact of those mutations. Of course, one of the most interesting things about the ROCO family of proteins is their modularity. With LRRK2, which is shown here in the center of the slide, you have the largest member of this family with its ROC and kinase domain. And we've shown very clearly, as well as many other groups, that the GTPase activity of LRRK2 is able to regulate the kinase activity, and subsequent work by a number of groups has demonstrated that the kinase activity of LRRK2 is able to feed back into the GTPase activity by phosphorylation. We were also very interested to learn how exactly this ROC core central domain of LRRK2 and the ROCO proteins were able to act as molecular switches, analogous to the RAS GTPases. And Joe Gibelli, a master's student in my laboratory at UCL at the time, was able to demonstrate using site-directed mutagenesis to get rid of GTP binding, that actually the GTPase activity of LRRK2, of DAPK1, acts to control the activity of the domain directly to its C-terminus, beginning to give us an insight into how these molecular switches work in a cellular context. Looking at cell survival as a readout for this, death-associated protein kinase 1 has been associated with cancers since its discovery back in the 1990s. We're also very interested in trying to understand the system that LRRK2 and its fellow proteins work in. And working with Mark Cookson over at the NIH, we've been able to identify a large range of interacting proteins, shown here as a spider diagram with LRRK2 over here in the center. And this begins to show another of the challenges that LRRK2 presents to us. It's a very gregarious molecule and has been reported to interact with over 400 proteins in the literature. Now, clearly, some of these are false positives, and there may be many which we're missing because they're not coming out either of proteomic screens or of pull-downs and so on and so forth. But it gives you an insight as to some of the challenges that we're faced with understanding the pathways which LRRK2 is associated with. This is something that myself and Claudia Manzoni and my group were very interested in looking at and discussed in a commentary paper in Science Signaling a few years back, thinking about the pleiotropic effects that we see with LRRK2 affecting multiple different cell types in inflammatory bowel disease, affecting the gut, in leprosy, affecting potentially immune response to bacterial infection, and in Parkinson's disease impacting on neuronal function in the brain. And we were really trying to understand how that could work, and looked at the possibility that, very similar to some of the other signaling pathways that have been very, very carefully characterized, for example, the mTOR pathway, what we might be looking at is a pleiotropic effect generated by differential expression in different cell types and with multiple interacting partners. And it's striking that this maps on quite well to the studies of pleiotropy at a genetic level carried out back in the 1930s and 1940s. This is a figure after Hans Grunberg, who was a geneticist at UCL back in the 1930s, looking at how individual genes could be linked to multiple pleiotropic effects in different tissues and different cells. And I think this goes to the heart of what LRRK2 is involved in, how LRRK2 is involved in all of these different disorders, and is something that we've been looking at in terms of cellular function for the past 6 or 7 years now. And to do that, we've looked at some of the proteins that LRRK2 is interacting with, and some of the vesicular processes in particular that LRRK2 has been implicated in. This really comes out of work from two groups back in 2007-2008, Charlene Chu's group over in Pittsburgh and Richard Wade Martin's group at the University of Oxford, implicating LRRK2 in the regulation and control of autophagy. This is a simplified diagram of the initiation and control of autophagy by mTOR and by the PI3 kinase Becklin pathway, where a phagophore is formed and is able to accumulate and clear waste from within the cell for recycling in the lysosomal system. Funded by the Michael J. Fox Foundation, we used a combination of genetic and chemical approaches to try and understand how LRRK2 was impacting on this cellular process, trying to move from a molecular biochemical study of LRRK2 through to understanding its cellular role. And using some of those inhibitors that I talked about earlier on in the lecture as a chemical tool to modulate LRRK2 activity, we were able to demonstrate that looking at a number of different markers for macroautophagy, in particular the lipidation and proteolytic processing of LC3, which sits on the outside of autophagosomes, we're able to show that the inhibition of LRRK2 kinase activity induced macroautophagy in these cells. And we're able to show that this actually increases autophagic flux by treating the cells with baflomycin to block lysosomal recycling of autophagosomes. We were, of course, intrigued as to what the pathways were which were actually regulating this. And so looking at the downstream readouts of the mTOR pathway, in particular phosphorylation of URK1, and later the downstream readouts of the Becklin pathway, we're able to show, using taurin as a control, inhibiting the mTOR kinase activity, shown here on the right-hand side, that LRRK2 is not acting to regulate macroautophagy through the mTOR pathway, and this has been replicated by a number of groups since. Interestingly, we do see a feedback loop, a long-term consequence on some of the downstream readouts of the mTOR pathway, in particular URK1, following chronic inhibition of LRRK2 kinase activity. And this is actually something that, to this day, we still don't quite understand exactly how this is occurring. Claudia Manzoni went on to look at this by targeting the Becklin pathway by using a combination of chemical modulators, including Wort-Manin and VPS34 inhibitors, as well as knocking down Becklin to try and understand whether this was acting through the Becklin pathway, and showed quite clearly that by removing and taking Becklin and PI3 kinase out of the induction of macroautophagy, that LRRK2 was operating through this pathway. So we began to build up a picture of how LRRK2 might be impacting on macroautophagy, not impacting through the mTOR URK1 pathway, but acting through the Becklin PI3 kinase pathway. And we're now beginning to move from a cellular understanding, and I think this very much speaks to a journey from the underlying biochemistry through to cellular function, and eventually through to physiological function in the system. And we're now beginning to look at all of those mutations that I talked about in the kinase and GTPase domains of LRRK2 in vivo, using C. elegans as a model. And collaborating with Eva Kavai at the University of Reading, who took up a lectureship at Reading a couple of years ago, we're now beginning to look at those readouts of macroautophagy in an organismal background using CRISPR-Cas9 genome editing to look at what happens when you insert those mutations in the endogenous C. elegans gene. And what we're now looking at are the readouts for autophagy in C. elegans. At the heart of all of this, and this has come out of work from a large number of groups across the world, most notably Dario Alessi's group at the University of Dundee, and also my former boss's group, Mark Cookson's group at the NIH, is a role for LRRK2 in vesicle trafficking. The work of my group has been very much focused on the role of LRRK2 in regulating macroautophagy, but there's now very good evidence that LRRK2 is operating to modulate shuttling of vesicles in the endosomal system between the trans-Golgi network and a variety of different cellular processes. And I think this begins to give us an insight as to how LRRK2 is able to have such a pleiotropic effect across a range of human diseases. And in particular, work from Dario Alessi's group and Matthias Mann's group over in Germany has identified the Rab proteins as direct phosphosubstrates for LRRK2 kinase activity. And this is an area of increasing interest across the LRRK2 field. I was attending a LRRK2 meeting over in Padova last week, where the groups from across the world were looking at how the alteration in Rab biology was correlating with disease mutations and with cellular readouts. And this was an area that I've been collaborating with Max Gutierrez at the Francis Crick Institute for about 6 or 7 years now. And earlier this year, Max's group published a study looking at what happens when you modulate LRRK2, both with inhibitors of kinase function, but also as a whole gene knockout in mouse models of mycobacterial tuberculosis. And that links back to the human disease situation and to the link between LRRK2 and leprosy. Leprosy, as many of you will know, is an extremely difficult disease to model in model organisms, and indeed some of the few organisms where you can grow leprosy bacteria is the armadillo, which is not an easily manipulable model organism. And so talking with Max a number of years ago, we decided to set in motion some experiments looking at what happens with the closely related bacterium, Mycobacterium tuberculosis, in the context of LRRK2, both genetic and chemical manipulation. What Max's group have shown over a course of some exceptional experiments in their mouse and cellular models for LRRK2 is that LRRK2 actually acts as a negative regulator of Mycobacterium tuberculosis phagosome maturation. And this again speaks to the pleiotropic role of LRRK2 in multiple disease pathways. And again, builds into this picture that's emerging of LRRK2 as a cellular controller of vesicle cycling. And it's notable that the phagocytic pathway and the encapsulation of Mycobacteria, it sits very much within the vesicular trafficking systems that LRRK2 is now being very, very closely linked into. So we've moved from the disease situation in Parkinson's through to understanding the biochemistry of the molecule, through to looking at the cellular consequences of LRRK2 manipulation. What does this all mean for Parkinson's disease? And so for the last few slides, I'll talk a little bit about some of the data that we have from patient material and patient cells suggesting that LRRK2 is acting to modulate autophagy and how this may be impacting on some of the pathological phenotypes that we see in the human patients who have these mutations. Working with a number of Parkinson's patients who carry mutations in LRRK2, very kindly donated skin samples to our group when we were working at UCL, we were able to look at the impact of LRRK2 mutations at an endogenous level and how they modulate the response to autophagy, building on the results using chemical inhibitors that we'd previously published. And what Claudia was able to show by inducing autophagy using starvation in these cells was that mutations across LRRK2 in each of the domains that are affected by mutations and impacting on each of the enzymatic activities of LRRK2, that mutations that cause Parkinson's disease reduce the ability of these cells to enter into macroautophagy. Again, looking at modulation of LC3, but also looking at P62 and WIPI2 as markers for macroautophagy. Working with Rina Bandapadai and Adam Maimas at the Queen Square Brain Bank at UCL, we're also able to look at this in human pathological samples to look at the end-stage pathophysiological consequences of LRRK2 mutations. And looking at readouts for macroautophagy, including LC3 and P62, we're able to observe, and Adam published this earlier this year, that actually LRRK2 mutations don't seem to be behaving in the same way that, or impacting the brain in the same way that is seen in idiopathic Parkinson's disease, i.e. those individuals who develop Parkinson's in the absence of a genetic insult. And what Adam was able to show was that in the brains of individuals with the G1219S mutation, which increases the kinase activity of LRRK2, that the accumulation of P62 as a readout for macroautophagy and LAMP1 as a readout for the lysosomal system, that these were differentially altered between LRRK2 Parkinson's and idiopathic Parkinson's disease. These are very challenging experiments to carry out. There are a very limited number of samples across the world of brain samples with LRRK2 mutations, and we're looking at the end stage of disease here. So you're looking at brain samples which are suffering huge degeneration, huge cell and tissue loss. And so these results should be treated with a degree of caution. I think it will be important to replicate these in larger samples. But this begins to suggest that there are disruptions to the autophagosomes and to the lysosomal system that are seen in LRRK2 brains that aren't seen in other cases. Adam was also able to show in a paper he published a few years earlier that actually if you look at the pathological readouts for Parkinson's disease, the accumulation of alpha-synuclein and the aggregation of this protein into Lewy bodies, that again there were differences between LRRK2 mutation carrier brains and idiopathic Parkinson's disease brains. And this slide really shows exactly how marked the impact of aggregation of alpha-synuclein can be in these brains. So in the middle column here, what you can see is a urea extract of alpha-synuclein from the brains of idiopathic Parkinson's disease, showing highly aggregated, very resistant accumulation of alpha-synuclein. This is shown by dot plot down here in the bottom panel. What we see with LRRK2 is actually that aggregated protein doesn't seem to be taking the same form. It is distinct from the aggregated alpha-synuclein that we see in the brains of individuals with idiopathic Parkinson's disease. And this was very surprising to us because actually these brains were very carefully matched in terms of the Lewy body pathology which is normally seen in these individuals. And I think maybe an indication of a fundamental alteration in the way in which LRRK2 is functioning in the brains of individuals with Parkinson's disease who carry mutations, and that may provide an insight into that pleomorphic pathology that I talked about at the beginning of this lecture. This provides a potential explanation through the degradation and accumulation of protein as to why individuals with the same mutation, with the same clinical phenotype, actually are very, very different when you look inside the brain and carry these different pathologies. And I think potentially provides an explanation as to how one protein can be involved in multiple different types of pathology, and may also speak to some of the alterations that we see in different disorders. And I think that the vesicle trafficking that is emerging in the LRRK2 field and its role in protein degradation and in the encapsulation of pathogens links into the underlying biology of LRRK2 and the physiological role of LRRK2 that has been the focus of my research for much of the last decade. Incidentally, the Rosetta stone was most noted for its ability to allow us to translate between Egyptian hieroglyphs, shown here at the top, and Greek, shown here at the bottom. And this, if there's, I don't know if anybody in the audience can read Egyptian hieroglyphs, but this is my attempt to translate between autophagy, which is a Greek derivation, into Egyptian hieroglyphs. And I don't know how well that stands up. I'll have to ask an Egyptologist as to how well that stands up. So during the course of this lecture, hopefully I've been able to give you an insight into the multifaceted nature of LRRK2 and its role in multiple diseases, multiple pathologies, multiple cellular pathways, but also multiple molecular events and multiple different types of enzymatic events within the cell. We're only just beginning to get the structural and molecular insights into how LRRK2 is functioning. It's hoped that within the next few years we will actually have a crystal structure for LRRK2. This is work that's been ongoing for a number of years now. And with those insights, we'll be able to build up an understanding of the cellular role of LRRK2, building that through to an understanding of how LRRK2 is functioning within organisms, and most importantly for the patients who have mutations in this gene, understanding how it impacts on disease biology and the development of treatments that can actually make a difference to Parkinson's disease. I started off by thanking all of the people that I've worked with. So at the very end, I'll just thank the funding bodies who have made this work possible, most notably Parkinson's UK for a senior research fellowship held at UCL and then at the University of Reading, the Medical Research Council for a new investigator research grant, and the Michael J. Fox Foundation, who funded much of the work that I've talked about. And thank you very much for your attention.